So I talked on Monday about just kind of the overall structure of cells, and that eukaryotic cells, which are really the focus of this course, are organized with internal compartments called organelles that have membranes around them. And so we could, I could spend a whole lecture or even a whole course on each organelle. Each one is kind of amazing and complex, and we know a great deal about each one. I'm going to focus on the nucleus today, partly because it's my favorite organelle because I work on it. It's also the organelle that holds the genome, and so it's the organelle that's kind of most relevant to the genetics and genomics components of the course. Kirsten says she can't unmute herself again, so I'm going to try to fix that, except I don't even see you in the list, Kirsten. Oh, wait. Okay, unmute. All right. No, you're still muted. I can't do it. Oh, there you go. Okay. We can look at the nucleus as sort of an example of an organelle. It happens to be the biggest organelle in most eukaryotic cells. Maybe the vacuole is bigger in plant cells. So it has its own structure and dynamics, and it has the chromosomes within it. And so I'm going to talk a little bit about chromosome architecture and nuclear architecture and how they are interrelated. At the very end of the last lecture, I didn't really have time to get to the slides about mitochondria and chloroplasts, which are also very interesting organelles, which I will not spend any time on. I just want to mention that both mitochondria and chloroplasts are thought to have been integrated into the last eukaryotic common ancestor had mitochondria, which were part of the emergence of eukaryotes, but mitochondria and also chloroplasts, which are unique to plants and algae, are thought to have come into cells through an engulfment process. They were their own organism, and both mitochondria and chloroplasts still contain fragments of a genome. They each contain a few genes that are needed for their own production. And that's part of how we know that their ancestral relationship to cells was that they were their own cell, and they came in and became part of the eukaryotic cell. So both chloroplasts and mitochondria have multiple membranes around them, which is also true of nuclei, which I will be talking about today. So that's all I'm going to say about mitochondria and chloroplasts, and you guys should definitely read that part of the first chapter just to refresh your memory about what they do and their organization. So the nucleus, as I said, is my favorite organelle. In animal cells, it's typically the biggest organelle. It tends to be located somewhere near the center of the cell for reasons that we'll probably talk about a bit later. It tends to be fairly round looking in cross section, although in tissue culture cells, they tend to look more like kind of like a fried egg. So although it's round in cross section, the nucleus is actually often quite flat. And this is a transmission electron micrograph where you can see the dark stuff here is chromatin, and I'll say more about that later. And this dark stuff is actually a nuclear body that's called the nucleolus, which hopefully I'll get to. It doesn't have membranes around it, but it forms sort of one or more discrete blobs within the nucleus. And there's a bunch of tinier blobs that you can't really see by electron micrographs that also form compartments within the nucleus. And I hope to get to that by the end of today's lecture. So the reason that nuclei are super important is because they contain the chromosomes, which is the DNA. It's where all the genetic information is stored. And the chromosomes, as we'll talk about in more detail toward the end of this section, go through cycles of replication, where they get copied, and division. And that division process is called mitosis. And the splitting of the cell is called cytokinesis. And so I'm not going to talk about these nuclear dynamics very much today. We'll talk about them when I talk about the cell cycle later in the course. I'm going to focus more on the organization of the nuclei when they're not undergoing division, which is most of the cell cycle. So when they're dividing, that's called mitosis, and the rest of the cell cycle is just called interphase, but it's actually when a lot of the interesting stuff is happening when most of the, when the DNA synthesis happens, or S phase, when most of the protein synthesis is also happening in the cytoplasm. And there's tons of transcription going on in the nucleus. So one thing I just want to remind you of is the fact that eukaryotes have this, you know, they enclose their chromosomes within a nucleus, means that the process of reading out the chromosomal DNA to make RNA and the process of reading out the RNA to make proteins is uncoupled. Transcription or RNA synthesis happens in the nucleus, and then the RNA is exported to the cytoplasm, which is where translation happens. That's different from bacteria, where it all sort of happens in one compartment, and transcription and translation can actually happen more or less at the same time. So that's a big fundamental difference between prokaryotes and eukaryotes. So chromosomes are large and beautiful, and you can actually see them during mitosis because they undergo a process called condensation, which I will not talk about in any detail today. But when they condense, they turn into these kind of thick fibers that you can see under the microscope, in this case through phase contrast microscopy, which is, so they're not stained, but we can also stain them with a variety of dyes and visualize the chromosomes. And they're very, that you can see them as individual entities here. This is from the textbook. Here's one chromosome going through this stage is called anaphase, when they're actually moving apart. And then this is an interphase nucleus, and all the DNA is kind of, it looks like it's all jumbled together. But as I'll talk about, there are kind of discrete architectural features that are kind of worth knowing about. So chromosomes were really one of the first fascinations for cell biologists. And that actually came about partly because in the late 1800s, there were a lot of, the chemistry was undergoing major revolution. There was a new middle class. The Industrial Revolution created a middle class, and the middle class wanted new stuff, and they wanted clothes that had bright dyes. And so there were dye manufacturers developing all these dyes, and there were people who, you know, had a lot of time on their hands and had microscopes, and that they were sort of playing with these dyes and putting them on cells and seeing what they stained. And so a lot of the dyes turn out to stain DNA specifically. Any idea why DNA might be particularly amenable to being stained with dyes? If you can think of an answer, put it in the chat. So chromosome just means colored body. And they were these colored bodies that people saw through the microscope, especially in response to being stained with certain, basic dyes. And Max said it's because it's negatively charged. That's right. DNA is an acid, deoxyribonucleic acid, so it binds very well to what are called, you know, basic dyes. DNA is called, it's basophilic. So it turns out that a lot of dyes wind up having chemistry that interacts well with DNA. And so people started describing what they saw, and they actually described in amazing detail the processes of mitosis and meiosis from a cytological standpoint, meaning like looking at the cells. And eventually people started to kind of connect the dots and realize that these bodies seem to behave in ways that might mean that they carried the genetic information that Mendel had described, and that the way that meiosis happened, which I will talk about toward the end of my lectures, seemed consistent with the way Mendel had described the segregation of genetic traits. So we still use a lot of dyes that were sort of discovered in a fairly random way to bind to DNA. If you take a pathology class in medical school, you will learn to look at sections like this. Typically, the nuclei are stained with a dye, often hematoxylin or other dyes, methylene blue in this case, that react with DNA or bind to DNA and light up the chromosomes inside the nucleus. In this case, these are blood cells, and the red blood cells are enucleated. Human red blood cells spit out their nuclei and don't have nuclei, so you only see nuclei in the other cells in the blood. Walter Fleming is credited with discovering these thread-like structures, which he named chromosomes, within the nuclei of cells, or the Kern, which is like the seed in German, and we've been studying them ever since. So Chromosomes, you've already heard a little bit about from, or maybe more than a little bit about from Gary. I'm going to have to be very superficial in describing chromosome architecture. Gary and I actually teach a whole course, a whole semester-long course on chromosomes and nuclear architecture. So this is kind of the Cliff Note version of that course. But just to remind you of something that Gary already told you, so the chromosomes are, you know, the DNA fiber within each chromosome is extremely long. The human genome is, you know, people always say 2 meters long if it's fully elongated as B-form DNA, just the kind of double helical structure that we're used to looking at. And so it has to be packaged to fit inside a cell nucleus, which is typically only a few microns long. So that's a you know, many thousand-fold compaction of this very long thread into a structure that has to be stable and able to replicate and divide without getting ripped up every cell cycle. These are just some kind of numbers for scale. This is, so chromosomes are enormous. In addition to the DNA, they contain roughly an equal mass of DNA and protein, which packages the DNA. And in addition, they contain sort of an unknown quantity of RNA, which is associated with physically with the chromosome. And that's something we know quite a bit less about. It's harder to study RNA. It's more labile and dynamic than the DNA or the protein. So we know kind of less about it. And I'm not going to talk about RNA associated with chromosomes at all today. This is kind of a typical kind of crappy textbook picture of how DNA is folded up inside the nucleus. This kind of idea of how DNA is folded up has undergone actually major revision in the last decade or so. I mean, you will often see these textbook images that show kind of hierarchical levels of folding, and that's no longer really the way we describe this. So I'll tell you a little bit more about how we think chromosomes are folded up inside the nucleus. So just to remind you, so every chromosome, at least every chromatid, every unreplicated chromosome contains a single, very, very long double helical DNA molecule. So that's what would be like 2 meters long if you extended the whole human genome in this form of B-form DNA. This is just the double helical structure. I am not going to talk at all about the backbone or the bases. If that is something you're rusty on, it's in the chapter. Please review it because it's kind of important core knowledge, but it's not really what I consider to be cell biology, so I'm going to skip over it. Okay, so this is just to give you a little bit more of a sense of scale of different eukaryotic genomes. Well, E. coli is not a eukaryote, but all of these are model organisms or humans. To give you a sense of the range of the size of the genome, this is in number of base pairs. So the human genome, and these are, I should point out, haploid genomes. So we're diploid, right? We have 2 sets of chromosomes, one from mom, one from dad. These are numbers for the haploid genome. So humans have 23 pairs of chromosomes. That's why 23andMe is called 23andMe, which means we have 46 chromosomes in every nucleus. The total number of base pairs in half of those, in the haploid set in 23 chromosomes is about 3.2 gigabases, 3.2 billion bases. And kind of on the smaller end of the eukaryotic spectrum is like the budding yeast, Saccharomyces cerevisiae, which has about a 12.5 megabase genome. So that's a pretty big size range. I think there are genomes smaller than this, and there are genomes actually quite a bit larger than the human genome, but this is kind of a useful range to think about. Chromosomes also have rather variable numbers of chromosomes. Bacteria typically have 1 circular chromosome, although there are many exceptions to that. Eukaryotic chromosomes are linear. They have ends that are called telomeres, and I am not going to talk about that today. I would like to, but there just isn't time. If you take 133L, you will learn about telomeres. They're the special structures at the ends of chromosomes that enable chromosomes to replicate themselves all the way out to the very end, and the telomeres also protect the ends of the chromosomes from undergoing fusion events. So that's about all I'm going to say about them. Okay, so the number of chromosomes varies quite a bit. and the length of DNA per chromosome. This is just the length of DNA in the genome divided by the number of chromosomes. And so you can see the range here. And then, it ranges over a couple of orders of magnitude, so quite variable. Okay, that's just for foundation. This is a terrible drawing, but I took it anyway. This is a mitotic chromosome. So this is a condensed chromosome, and it's replicated. So it has this X-shaped structure because there are two chromatids, the two copies of the chromosome. And in the middle here, they're held together, and I'll talk more about this again when I talk about mitosis. They're sort of stuck together in the middle where this structure called the centromere is. So the centromere is one of a number of different functional elements that are contained within eukaryotic chromosomes. And I'll have a lot more to say about centromeres when we talk about mitosis, but centromeres are the part of the chromosome where the kinetochore assembles. The kinetochore is this structure, rather complex kind of plate-like structure that enables the chromosome to attach to microtubules, which I haven't talked about yet, which is why we're going to talk about mitosis later. But microtubules pull the chromosomes towards the poles during mitosis and meiosis, and the centromere is the specialized region of the chromosome where that happens. The telomeres I've mentioned are sort of like caps on the ends of the chromosomes that enable them to replicate and protect them from undergoing aberrant fusions with each other. And then scattered along the chromosome are also sites that are called replication origins. And I'm not going to talk about replication really at all. Again, this is something that I consider to be more of a topic for a molecular biology course. If you are unfamiliar with replication, I suggest that you review it. But replication origins are, they're different in different organisms, and they are the points where that process of replication tends to initiate. And there are multiple points of initiation along the chromosome, so the replication forks move out and eventually replicate the entire sequence of each chromatid, and to make the two chromatids that will then separate in mitosis. Okay, so I showed you that we can look at chromosomes by phase contrast microscopy. There you can really only see them when they're condensed in mitosis. We can stain them with a variety of dyes. The most common dyes that we use now are fluorescent dyes. So you might hear of Hoechst or DAPI. Those are two different fluorescent dyes that bind to DNA and kind of light up the nucleus. They're used almost all the time in fluorescence microscopy. There's also a technique called in situ hybridization, where we use probes, which I'm not going to explain. Again, if you take 133L, you will learn about how this works. But you can design probes, which are made of DNA, and they hybridize, they stick to specific sequences in the genome. And by designing them in different, and labeling them with different dyes, you can actually design probes that will light up each chromosome in the genome. They're actually sort of combinatorial colors, but for the purpose of explanation, we'll just say they light up each chromosome in a different color. And so you can take, if you hybridize, if you stain a sample with these probes, you can actually then computationally, you can kind of cut out and make an image that looks like this, which is we call the karyotype, or the chromosome complement of a particular cell or organism. In this case, this is a normal human female. Whoops, sorry about that. So there should be 46 pairs of chromosomes, 2N, so N is 23, 2N is 46. You'll see different kinds of designation. This means that there are 44 autosomes and 2 X chromosomes here. There are the 2 X chromosomes. That's how we know this is a female. The chromosomes in mammals are typically numbered roughly in descending order of size. So chromosome 1 is the biggest, 21 and 22 are very small. Okay, relatively speaking. So let's see. And Max asks, to clarify, is a probe DNA that can hybridize to the chromosome in question plus a dye? Yes, I think that's a good way to put it. Okay. Okay, so chromosome numbers can change. Obviously, like mouse, I told you, has 20 pairs of chromosomes. Humans have 23. Clearly, but in the evolution where mouse and human bifurcated, something happened to change the number of chromosomes. That normally happens on a fairly fairly slowly. So closely related species will often have the same number of chromosomes, and they will have essentially the same genes on the same chromosomes. But there are cases in evolution where massive rearrangements happen. And this is a particularly striking example that's in your textbook of two very closely related Asian deer species, the Chinese muntjac deer and the Indian muntjac deer. They look pretty similar. Their genomes are actually pretty similar in overall size, like number of base pairs, but something crazy happened along the path that led to these two species. This one underwent massive chromosome fusions for reasons that I don't think anyone knows. And this one was probably more like the ancestral state. So this deer has 23 pairs of chromosomes, and this deer has 3 pairs of chromosomes. It has 2 Y chromosomes, which is a little unusual, but not very unusual. Sex chromosomes are incredibly plastic. It's a whole topic unto itself. You'll also notice maybe that these chromosomes are sort of different in structure. These kind of look like wishbones, and these look like X's. These are, again, mitotic condensed chromosomes. These look like wishbones because the centromeric region where the sister chromatids are held together is near one end, and that's called acrocentric. Whoops, sorry about that. Whereas these chromosomes have their attachment point more in the middle. Again, that's the centromeric region, and by definition, those are metacentric. You can see this Y chromosome is not metacentric. This one's kind of off. So it's got one big metacentric chromosome, one, I guess, more acrocentric chromosome, and some weird stuff going on with the sex chromosomes. So we don't entirely know what drives these kind of rapid changes in chromosome organization or complement, but chromosomes can undergo fusions and they can break apart. When that happens within a species, it usually is bad. It leads to infertility. If you have exchanges between chromosomes, it can create oncogenes. It can create, we'll talk more about that later, but it can put two things together that don't belong together and lead to aberrant expression of things. So normally the goal of the cell is to maintain genome stability, not to allow chromosomes to undergo rearrangements or too much mutation. But it does happen in evolution, and if it's tolerated, then you can wind up with a situation like this where you have closely related species with very different karyotypes. It can also happen in response to DNA damaging reagents. The best known of which is radiation. Actually, Muller won back in the early days of genetics. People didn't know how to make mutations on purpose. They just had to wait for them to happen spontaneously. And Hermann Muller was the person who discovered that radiation can damage DNA and induce mutations. And so this is a chromosome spread from a person who was affected by the fallout from the one of the two worst nuclear power plant disasters ever, the other being the one in Fukushima more recently. But this was a huge event during my kind of adolescence, where there was massive radiation and many people developed cancer. And that was largely as a result of mutations and chromosome rearrangements arising from the radiation. Okay, so by using these same kinds of probes, these like in situ hybridization probes that we can use to identify specific chromosomes in a spread of condensed chromosomes like this, we can also look at the organization of chromosomes inside the interphase nucleus when they are decondensed. And so if you just stain with a DNA dye, it stains everything, not quite homogeneously, but it stains everything. You can't tell where individual chromosomes are. But if you use this kind of painting probes, you can actually see where each chromosome is within the nucleus. And this technology has enabled us to study many aspects of how chromosomes are organized within the nucleus. This is a very big field, and I'm just going to highlight a couple of points about it. But so one thing that was discovered early is that chromosomes tend to occupy what we call territories. They're not spread throughout the entire nucleus. They are sort of concentrated in blobs within the nucleus. So this is chromosome 5. Here's the other copy of chromosome 5. Here's chromosome 11, and so forth, the 2 copies. Here's 3. And what people have learned over time is that although there is a lot of randomness. So like chromosome 5, here you can kind of tell this copy of chromosome 5 and this one, they look pretty different. You know, they have different shapes. This one looks like it's up against the nuclear envelope. This one's more in the middle. So there's a certain amount of randomness, but there are also biases. And so like a well-known one is that this is a kind of well-known paper from just before 2000, where this shows two different chromosomes. These chromosomes were picked because they're similar in size, but chromosome 19 has a lot of genes that are expressed in this tissue, and chromosome 18 is much less expressed. So there's a lot more transcriptional activity from this chromosome than this one. And what was observed in this paper is that the chromosome that has much less expression more often is associated with the nuclear envelope. And I'll tell you a little bit more about that, about why that is in several slides. So there's random, but also some bias. Was there a question?
Yeah, Abby, someone has a question. If this is true of most replications.
What does that mean? I don't know what they mean by replications.
Yeah, I'm not quite sure either. Naomi?
Naomi, if you can clarify, that would help. Yeah, we'll come back to that. Okay, so how do chromosomes, how does this incredibly long strand of DNA get packaged up into a territory? So this is still a topic that's very highly, under very active research, and there's a lot of controversy. I mentioned earlier that you tend to see these kind of hierarchical diagrams in many textbooks. A lot of these are going away now because this, so this is how I learned it, right? There's the DNA fiber, the double helix. This is not controversial. The DNA, the first level of packaging is that DNA is wrapped around these structures called nucleosomes that Gary talked about, and I'll remind you a little bit about what they are. And then for many years, people have argued about whether there's some kind of intermediate structure. People thought that they saw this structure so the nucleosome fiber is about 10 nanometers in diameter. People observed like frequent fibers in the nucleus that seemed to be about 30 nanometers, and they speculated they might have some kind of regular organization. This is still very controversial. I tend to fall on the side that there is no specific 30 nanometer fiber structure. Let's see. So Naomi said, in S phase, are the most expressed chromosomes replicated first in most organisms? That's a really good question. So yes, the highly expressed chromosomes tend to be more what we call euchromatic. They tend to be more active and more accessible, and the same packaging features that make them accessible to the transcriptional machinery also seem to make them accessible to the replication machinery. So they tend to replicate earlier. And that's also true on the subchromosomal level, like regions that are highly expressed tend to be replicated earlier. So there's a strong correlation between early replication and high levels of transcription. That's a really good question. Okay, so all right. So we do think that this doesn't give nearly the degree of compaction that we know has to exist within the nucleus, and so there's just a lot of controversy about exactly how that compaction is achieved. But it is now, I think, widely accepted that chromosomes are folded into loops, and that those loops are formed probably by proteins that act as motors, and I'll show you what I mean by that momentarily. And then the loops undergo kind of another kind of level of packaging into what people have described as like a helical staircase to form the mitotic chromosome, which is even more compact than the interphase chromosome. So I'm not expecting you guys to like know how this works, because nobody really knows how it works. Like I said, it's still a very active area of investigation, and I'm happy to give you guys some references if you want to see where the state of the field is. Okay, so nucleosomes, just to remind you, hopefully, are each little bead. We call this a bead on a string structure. So the beads are made of protein, and the proteins are histones, which are these tiny, very, very small basic proteins. So DNA is acidic, the proteins are basic, and they're attracted to each other through electrostatic interactions. And there are 4 what we call core histones. H2A, H2B, H3, and H4. And these kind of self-assemble, these proteins self-assemble with the help of additional proteins that are called chaperones that help them interact in an orderly way. They form these nucleosome structures. and the DNA wraps around the nucleosomes, not quite 2 turns per nucleosome. And they create this beads on a string structure that we call the 11 nanometer fiber. And this just shows that there are nucleases, often micrococcal nucleases often used, that actually tend to digest DNA between the nucleosomes, so we can actually sort of map where the nucleosomes are along the DNA sequence. Okay, so Gary talked about this, I think, at length, and I'm just going to remind you, but it is super important for understanding chromosome structure, so I didn't want to like just rely on what Gary had said. So the histones form this bead-like structure. That's and the histone proteins themselves have what we call the core, which is the part that's part of this bead-like structure, and then they also have these N-terminals. So at the N-terminus of each of the histone proteins, there's this kind of flexible tail, people call it a tail, that sticks out from the bead particle, the nucleosome particle. And those tails are extremely rich in amino acids that can be modified by other enzymes, especially lysine residues. You'll know, so this is the tail of histone H3. Histone H3 and H4 in particular undergo massive, very dynamic modification by other proteins, by enzymes. So lysine is basic, arginine is also basic. These are reactive residues that can be modified by the addition of methyl groups, acetyl groups, additional groups like citrulline and other things that are kind of more exotic, but these are the best studied, methylation and acetylation. Typically, a particular lysine can be either acetylated or it can be methylated. It can't be both at the same time, but it can have 1, 2, or 3 methyl groups on it. So you'll see like notation that's like trimethyl, dimethyl, sometimes it's just written as Me2 or Me3 or something like that. And so Gary talked about the writers and erasers that put down these marks on the histones. Those control the association of additional proteins, which control basically the packaging of specific regions of the genome. And there's certain, particularly well-studied modifications. They're not necessarily more important than others, but they're very well studied, that have specific properties. So lysine, so K is lysine, lysine 9 trimethylation recruits heterochromatin protein 1, and that leads to a particular kind of packaging of the DNA. Conversely, trimethylation of lysine 4 on the same histone, on histone H3, is associated with very active genes. So this is repressive. So it tends to silence genes from being expressed or replicated, and this activates them. So methyl is not inherently silencing or activating it, but it's where it is on the histones that matters. Okay, and right, so there's a ton of different pathways that modify these histones on either a very local level or sometimes on a much broader level. And so,
yeah, Abby, there's a question.
Yeah, condensed mitotic chromosomes. Yes, mitotic chromosomes also contain tons of proteins, and there is no histone attached during interphase. No, the histones the histones are there all the time. These nucleosomes are present all the time. They do get moved aside transiently as the chromatin is being transcribed, but then they reform again, and so there's so roughly half the weight of a chromosome is histones. They're probably a little less than that, but they are the most abundant proteins, and they are present in chromosomes throughout the cell cycle. They get modified differently during mitosis, but they're always there. Gary talked about how when chromosomes replicate, the nucleosomes also have to be replicated, and so they get doubled as the chromosomes are replicating. Trimethylation and acetyl both require, yes, they're like the whole kind of system is required for expression. Yes, it's true that acetylation is associated with active expression, but as I said, it really depends on which residue you're talking about. So, okay. So this is more of a space filling model of the nucleosome. So this was a structure that was solved by X-ray crystallography, and it shows kind of the way the DNA is wrapped around the nucleosome. And so that, yeah, and this just highlights where the different histones are within this core particle. And again, it's an octamer, so that means there's 2 of each of these 4 histones. Okay. and I'm sorry that I know this is a rather superficial view of a very complex system, but I'm trying to highlight the things that I think are most important for understanding the nucleus and the architecture, and not so much focused on the intricacies of individual gene expression, because that's sort of a topic for a different course. I'm really more focusing on what I consider to be the cell biology of chromosomes, just like their spatial organization, and their interaction with other features in the nucleus. Okay, so this kind of shows very, very schematically, kind of imaginatively, I would say, the overall folding of a whole chromosome, just highlighting that different parts of the chromosome, the telomere, the centromere, other regions of heterochromatin, that there can be big blocks in which the specific marks like trimethylK9 is highly enriched, and that's true in what we call the pericentromeric heterochromatin, and often adjacent to the telomeres. And the telomeres and centromeres themselves have special histones. Well, actually not the telomere. The centromeres have special histones. I don't know if Gary talked about SEMP-A, which is a special version of histone H3, which is localized to the centromere. Anyway, so there's also histone variants and so forth. But so the histones, the way that they're modified then in turn kind of leads to differential packaging of different regions of the genome, which controls which genes are expressed in different cells. And yeah, and that can happen, as I said, on local, like gene by gene, or even within a gene level, but there are also big blocks they're big regions of the chromosome that can be kind of modified to be more heterochromatic and so forth. Okay. Gary talked a little bit about this. I'm not going to go into any detail, just that some of these marks, and he probably talked about H3K9 trimethyl, which is a silencing mark, they are thought to actually spread along the DNA in a sort of linear way through feed-forward mechanisms. And that's because the mark, so once the mark is there, it actually recruits the enzyme, the histone methyltransferase that makes the mark. And so the presence of the mark can lead to adjacent nucleosomes being modified and lead to that mark spreading along the chromatin. But there are elements in the genome that act as barriers to that spreading. And we don't entirely understand how those work, but they sort of limit how far that spreading will go. So as I said, like on a smaller scale, you can have like a series of genes along the chromosome, and whether or not a particular gene is expressed will be reflected in its packaging. I don't want to say the packaging controls it, but it kind of does. So this cell might have transcription factors that bind, upstream of genes A, B, and D, and open up the chromatin and enable it to be expressed. And this gene C might not be expressed. I mean, here it's showing being expressed, but imagine it's not. So if you map the histones across a gene that's not being expressed in a particular cell type, it would look more heterochromatic than these genes that are being expressed. So there's interplay between the transcription factors that bind to the DNA and determine whether a gene is going to be read out, and the histones. They kind of affect each other. Histones can prevent binding of histone modifications can prevent binding of transcription factors, but transcription factors can also influence the packaging of the gene. Okay, and I said that we now think that the 11 nanometer fiber, so that the histone beads on a string structure, which isn't really shown here, but that's what this is indicating, is then folded up. And for a long time, we just didn't have any idea about how it was folded up. And now people really think that the main proteins that fold up the genome are these proteins called cohesins and condensins, which I'll say more about in a later lecture. But they are thought to actually walk along DNA like motors, and they may work in pairs, so that this is just a model. It's very controversial, but they may work in pairs where they're kind of walking along the DNA, and in a pair, they can kind of extrude a loop of DNA or chromatin. And it's thought that that's kind of how the genome is folded up. The evidence that this happens, is really only a couple of years old, and it was very controversial until there was kind of direct experimental evidence. So there's a very active area of research. And I'm not going to expect you to like understand the kind of all the nuances of what people think, but it is good to know that we now think that these loops are major units of DNA, and these are dynamic. So it's a static image, but these things are kind of moving, and that the points where these loops kind of there are points in the genome that bind to specific proteins that tend to stop. We think they stop the motors from moving, and so it's not all or nothing. But these are, you can think of these as like pause points. And so if you look at a snapshot of how the DNA is folded up at any particular moment in time, there are regions that are more likely to be the base of loops than others, and these loops can all loop kind of barrier activities also act as barriers to the spreading of histone marks. So they sort of create these domains of chromatin that can behave in a somewhat unified way, but they're not static. Okay, I know that's very confusing. I'm happy to talk to anyone who's interested about this in more detail, but I want to get kind of a little bit farther today. Okay, so Gary also, I think, talked about a case where you can get very broad spreading of histone marks, and that is something that happens in the process of dosage compensation. So he talked about sex-linked genetic traits and how you can have, in a female mammal, for example, you have 2 X chromosomes, and what happens during early development is that there is a random silencing of one copy of the X chromosome in each cell. So in this case, the paternal XP X chromosome becomes silenced, the other one remains active, but it can happen the other way around, and it's a random process. And the inactive X chromosome in each cell becomes heterochromatinized. It becomes enriched for H3K9 methylation and a bunch of other marks, which I don't expect you to remember. But the net result is that the chromosome is greatly suppressed for transcription. It's not completely silent, but most genes are silent. This is shown with the silent chromosome up against the nuclear envelope because it often is. There are proteins in the nuclear envelope that interact with heterochromatin, and so heterochromatic regions tend to be at the nuclear envelope. I showed you that with the chromosome 18 and 19 earlier, where one is more active than the other, and the more active one tends to be in the middle of the nucleus, the less active one tends to be at the periphery. This is kind of an extreme case where this chromosome is inactive, and the heterochromatin interacts with proteins that bind to the nuclear envelope, and so it tends to be at the nuclear envelope. And that's shown here. This is a kind of extreme way of staining it, where you can really see the structure that is known as the bar body. That's the inactive X chromosome, and it's often kind of plastered against the nuclear envelope. We don't necessarily think it has to be at the nuclear envelope to be silenced, but we know that the nuclear envelope has proteins that interact with heterochromatic proteins. Okay, and that silencing process, I know Gary talked about calico and tortoiseshell cats and how they have these patches of different color that reflect silencing of the maternal or paternal chromosomes. So I'm just reminding you about that. Okay, so the nuclear membrane, we have like less than 5 minutes left to talk about a rather complex structure. So the nucleus is surrounded by a double membrane, an inner nuclear membrane and an outer nuclear membrane. And the outer nuclear membrane is contiguous with the endoplasmic reticulum, which I will have more to say about when I talk about protein trafficking. So that's a membranous material that kind of extends throughout the cytoplasm, but it's actually contiguous here with the outer nuclear membrane, which seems really confusing and doesn't make sense, but just accept that that's how it is for now. And this is a crazy picture of chromosomes. I hate this picture, but anyway, but I like this picture of the nuclear membrane because it highlights a few useful things like this, that the outer membrane is contiguous with this endoplasmic reticulum. These are ribosomes, by the way. And then the nucleus is studded with these structures that are called nuclear pores. And that's how things go in and out of the nucleus. I will talk more about that during the protein trafficking lecture as well. But this is what they look like. They literally are like little rivets through the 2 membranes of the nuclear envelope. And you can see them by different EM techniques. This is a TEM, transmission EM. They have this 8-fold symmetry. They're made of between 30 and like 60 proteins, depending on which organism we're talking about, and they self-assemble to form these donut structures with holes in the middle. They're not empty in the middle, they have this kind of goo in the middle that regulates the passage of things in and out. And I'll talk more about that hopefully later too. But this is just a diagram. They're asymmetric. On the cytoplasmic face, they have these kind of little projections, and then they have the structure called a basket on the inner side where the chromatin is. The other thing I wanted to highlight is that in metazoans, so in animals and their relatives, the inner nuclear membrane has a layer of protein under it that forms a meshwork called the lamina, the nuclear lamina, and it's made of nuclear lamins. And I will show you that in a second. So here's just another image of the cytoplasmic and the nuclear face of the nuclear pores, showing this structure that we call a basket on the inner side. And they're made of proteins called nucleoporins, or sometimes just abbreviated NUPs. So here's another view. Here's the nuclear pore going through the outer and inner nuclear membrane. And this structure here, which is just drawn as like scribbly lines, is called the lamina. People have found ways to kind of expose it on, make preps where the nucleus is kind of opened up and do like electron microscopy on it. This is probably not a great representation of what it looks like, but it shows that it forms like a cross-linked basket-like structure. So these lamin proteins make something called intermediate filaments. I hate that term because like, intermediate between what and what. I think it's between actin and tubulin in diameter, but we'll come back to that when I talk about the cytoskeleton. Anyway, these proteins self-assemble to form this meshwork. It stabilizes the nucleus, and it also interacts with the chromatin, as I'll show you in a sec. And I'm mentioning it partly because mutations in the protein, in the lamin proteins that make up this meshwork, or in some of the associated proteins like emerin, cause a number of human diseases. It was really striking when a mutation in lamin was found to cause a particular kind of muscular dystrophy. And emerin also, mutations in emerin can also cause this Emery-Dreifuss muscular dystrophy. So we don't actually know entirely why these diseases arise, if it has more to do with the structural integrity of the nucleus or the or gene expression defects that are associated with mutations in these proteins. And yeah, so I'm gonna not talk about the nucleolus right now. I'll pick up with that on Friday, but I just wanted to mention that I already sort of alluded to the fact that certain kinds of chromatin, particularly H3K9 methylated heterochromatin, which also looks kind of dark by electron microscopy, it interacts with proteins that are associated with the nuclear lamina. So it's not 100% at the nuclear lamina, but it's enriched at the nuclear lamina, which is why you often, like if you look at animal cells or metazoan cells, the edge of the nucleus is enriched for this dark staining heterochromatin. Okay, so it is already 10, and I knew I probably had a little bit too much to get through today, but I'll pick up on Friday and talk about nucleoli, which are these kind of bodies within the nucleus where ribosomes are assembled. And I want to talk about that partly because later I'm going to come back and talk about organelles that don't have membranes around them. And the nucleolus is a really nice example. So I'll start. And yes, yes, we should have a GSI meeting now. Before we do that, I want to ask if there are any questions. I know this was kind of dense and I apologize, but I couldn't really cut any of this out. I'm going to unmute you guys. Well, actually, that's probably a bad idea because there's too many people on. So if you have a question, type it. Do the histones stay on DNA during replication? Like during transcription, actually, that's something that Carlos Bustamante's lab at Berkeley studies. I don't remember exactly what happens during replication. I think they may split. I don't think they come off entirely, but I think they may open up and get remodeled. What does it mean for chromatin to be enriched? Well, okay, what I was talking about there is that, whoops. So chromatin is throughout the nucleus, and what I was saying is but there's enrichment. Enrichment just means it's concentrated. That that the heterochromatin, so the terms euchromatin and heterochromatin were based initially on just the way that they stain by both fluorescent dyes or other kinds of DNA dyes, or by this kind of electron microscopy. So the heterochromatin stains more heavily with heavy metals when you stain nuclei for electron microscopy. And it's still controversial if that is because it's more densely packed, or if just the proteins that are associated with heterochromatin tend to like the metals more, and so they pick up more of the metal stain. But heterochromatin stains darkly, and by enriched here, I just mean that the dark stuff, this is the nucleolus, so ignore that for now. This darkly staining heterochromatin is enriched, meaning it tends to be at the nuclear periphery in most mammalian cells. There are some really interesting exceptions. I don't know if that answers the question, but I hope so. If not, ask me separately. Anyone else? Let's see, which part of chromosome is more likely to be affected by heterochromatin mediating silencing and expression, since there are heterochromatin on both the centromere and telomere? Okay, so yes, the telomere itself does not have genes within it. The centromere itself, well, I don't want to say it, the centromere itself can be a big region, and there can actually be some genes kind of in it or near it. But those regions do tend to be silenced. It's complicated because there are genes that are actually adapted for being expressed when they're in heterochromatin. So the book has this bit about how people have studied the effects of telomeres, particularly in yeast, in budding yeast. If you move a gene and stick it right next to a telomere, it will tend to be silenced because the heterochromatic proteins associated with the telomere actually kind of spread into the gene and make it harder for it to be expressed. I don't know if that, let's see. But in general, genes are that they are in a place on the chromosome where they can be expressed. So if a gene wound up getting stuck somewhere it couldn't be expressed, that would be probably a lethal mutation, and so that would not happen. You would never see that. We basically see, when we look at existing organisms, we see what has worked in evolution. And so, the genes on the chromosome are all in places that they can be expressed appropriately. I don't know if that explains it, but so the heterochromatic regions like the centromere and telomere tend to be, they don't tend to have genes in them or many genes, and the genes that, if they are there, they have some special mechanism that enables them to be expressed in that context. Okay, no more questions. Maybe, I think the GSIs, I will just send you a separate link so we can Zoom each other. I'll sign off for now and see you guys on Friday. Nobody came to my office hour yesterday. I was so sad because I was like, how am I gonna get to know you guys since I can't even see you? Anyway, come to my office hours just to say hi or not, but I do miss having actual people to talk to. This is a little strange. Anyway, all right, goodbye for now. Let me know if you're having problems accessing either the materials or the textbook or anything else, and we'll try to keep things going. All right, take care. I can't turn it off.